Hi, everybody. Thanks for coming. And for those of you out there in YouTube land, hello, now and later, I suppose. My name is Alan Andrews. I'm a research fisheries biologist here at the Pacific Island Fisheries Science Center. So I work for NOAA Fisheries, the federal government. My position is in the Life History Program with the Fisheries Research and Monitoring Division. Specifically, I look at life history traits. I try and figure out how long fish live. So like, what is their longevity? How quickly do they grow? What is their age at maturity? Not just length at maturity, but age at maturity. And all of these questions come together for information that is very important for sustainability of the population. We have to know how it grows in order to manage it properly. So tonight I'm going to introduce you to some work I just recently completed on the Blue Marlin, which is sort of an elusive fish in terms of figuring out how long they live. I finally came up with a way to do it with some colleagues here. And this is going to touch on quite a bit of work that others have done over the years. So I'm sort of standing on the shoulders of giants in many ways. You know, a lot of this research has got a long line of background that comes from people that have also done a lot of work. So I'm going to carry on the tradition, but with what I'm going to share with you today. So we're going to look at how nuclear bombs can help us validate marine organisms. So these awful things that we blew up in the atmosphere actually helped us out in a little way. And specifically, I'm going to talk about this fish. This is a Blue Marlin. It was 1,245 pounds, 3.7 meters long. So it was a very big fish, and it was actually captured just south of Honolulu, which is a little bit rare because typically the big marlin are brought in to Kona. The little inset there that you can see, and I'll try and use the pointer here so everybody can see it. These are the otoliths. So these are the fish ear bones of this fish. Now they were only 8.1 milligrams. That's the edge of a US dime that you see there for scale. So they were very, very small. Not much material to deal with. Typically, I deal with a couple of grams worth of material, or at least tenths of grams worth of material in order to do some kind of chemistry analysis where we can answer questions about, hey, so we can answer questions about how long these fish live. So I'm going to step right into it. So age estimation typically deals with looking at hard parts and looking at growth increments in those hard parts. So zones that might be annual, sort of like what you see in a tree, right? And with this species, they investigated the vertebrae. They were pretty much useless. Fin spines, the dorsal and anal fin spine actually were a little bit helpful. They came up with increments and estimates of age. And then they also looked at the surface structure of some of these small otoliths. And otoliths is the ear bone. So that's what I'm talking about, the otoliths I showed you at first. Otolith is the fish ear bone. So they noticed these little increment structures on it. Well, you know, maybe that's the analogous 1 per year growth band structure. And if you look over here on the side, this is the growth curve that they came up with. So here's a fish that's like 4 meters long, is maybe on the order of estimated to be about 20-something years old. Okay, you see that? So follow up with this. People have also looked at the otoliths, looked at the outside, but this is actually a section of the otolith. So if you look here, if I can get the cursor to show up, you can see there's a band structure here that's very, very similar to the structure that you see on the exterior. And so in general, across the board, most of the studies have come up with age estimates that are on the order of 20 to 30 years. Here's the most recent work done by some Japanese researchers, Tamaki Shimose and others. They validated the earliest growth, and they looked at this very, very fine increment structure, not annual, but daily. So during the time when the otolith is growing the fastest, you can actually see daily increments. And so you can validate the first year and maybe 2 years worth of growth. But as you get older in life, the growth zones become far too compressed and you can't count them anymore. So it's only good to validate early stuff. So they came up with also, they looked at annual growth zones, and here's the growth curves that they got for it. So notice, so here's fish length on this axis here, and then here's estimated age from counting these band structures that they see. And once again, you see it gets out to about maybe 20 years. Some grow really fast, maybe 3.5 meters in 8 years. This one's maybe around 20 years and is smaller. So it looks like there's a lot of plasticity in the growth too. They can get big quick, but maybe they can stick around a little longer. So, and the daily increment stuff, it actually corroborated some work that was done early on by Eric Prince and others in 1991. They get to be 1 meter to 2 meters in the first year to 2 years of life. So a meter's like 3 feet, right? So that's first year of life. And so when that first paper came out in 1991, people pretty much poo-pooed it. They didn't think, no. But turns out that Shimose actually showed it also. Yeah, did you have a question?
Each one is a fish?
Each one, yeah, so each one of these dots is a fish, okay. Right, yep, yeah, for a certain size and age estimate, right. So you use a population, a sample of the population to try and describe the growth characteristics of the group of fish. Okay, so the max age so far is about 20 years. Now, if we look at all of these things, typically we get an age of about 20 to 27 years, depending on the study that you look at, but validation is necessary. This is just counting growth bands. We're assuming that the bands we're seeing are 1 per year. So I'm going to take a step towards a fish that's local, the onaga. I'm going to show you how a method that I use called bomb radiocarbon dating works by using sort of a best case scenario with a local snapper. So this is the onaga. It's a deep water fish. If you've ever had it, it's quite tasty. Gets to be close to 1 meter long, and it's bright red. It's really beautiful. Here's their otoliths. So this is the fish ear bone. The top one is a fish that's estimated to be about 2 years old. The bottom one is the oldest fish and our largest fish that I had. And what we do is we section these otoliths. And here's what we do. So this right here, so this is the otolith. If you section it like this right across here and then lay it on its side, see all those growth band structures there? It looks like a nightmare if you try to count it, right? It's pretty tough to do. But once you develop a search image of age reading lots and lots of otoliths, then you start to go, oh, okay, here's the pattern. And notice that there's an attenuation of the growth. Most rapid here, drops off. I don't know, can you see those little tiny spots? I probably need to make them bigger because I'm going to publish this eventually. I wish this would not go away. So there we go. So see, they get smaller, 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 and it goes all the way out. And if I counted this a bunch of times, I got pretty close to 55 years for each read on this particular fish. Now, it turns out that my biggest fish actually happened to be one of the easiest to read, and I called this my Rosetta Stone. I wish they all looked like this, but they don't. So I tend to refer to the search image that I get from these really well-defined otoliths. It's like, okay, this one looks good. It makes sense. And I try and count the other ones that way, right? But again, the question is, are these growth zones annual? They're very, very fine in structure. Who knows? Maybe it's 2 per year. Am I overcounting? Am I undercounting? We don't really know. So in steps this amazing technique called bomb radiocarbon dating. It relies on atmospheric testing in the 1950s and 60s. When we reached thermonuclear levels, okay, once we got into the thermonuclear devices, we effectively doubled the radiocarbon signal on the planet. So we sent off a lot of other stuff, strontium-90, cesium, iodine, a lot of other nasty elements and isotopes. But radiocarbon, we actually doubled the amount that was naturally existing. So there's naturally existing radiocarbon levels, but we effectively created a spike right on top of that. And here's what it looks like. I'm kind of skipping over the atmospheric record. So once it goes into the atmosphere, so these thermonuclear devices went into the stratosphere, created a global signal as it worked its way back into the troposphere. Then it diffused into the oceans. And this is the record from corals. Okay, so corals, we can count the growth bands, right? We know or can validate that these growth bands are 1 centimeter per year using another technique. So if we look at the radiocarbon content across these bands, this is what we get. So you see, here's pre-bomb. Okay, so let me explain this a little bit here. So see, this is delta C14. Now, delta just means difference from a standard. Okay, it doesn't mean 0. So these levels are lower down here because we're in the marine system. So it's right at about minus 50 or so pre-bomb. So you get about 1954, 55, it starts rising. And then 1963 is when the treaty was signed, no more atmospheric testing. A little bit of, it's still going to go up for a while because it's working its way into the atmosphere and into the sea. So there's a phase lag, and then post-bomb peak decline after that. Okay, so we've got kind of a time-specific marker. And if you look at this, one record, the first record is Curie Atoll, which is way out in the Northwest Hawaiian Islands, all the way down to Kona, the Big Island. So it covers a pretty big swath. And it's kind of surprising, if you look at it, that the ocean chemistry is that similar in its timing. And that means it's a great tool for aging other carbonates, because this is carbonate, right? This is carbonate. This is formed from the CO2 that diffused into the ocean. It created calcium carbonate from the radio CO2. Okay, so, and let me know if you have any questions. I'm totally flexible. You can just raise your hand. Yeah, you're good. Okay. Okay, so that's the coral. So now we come back to the fish, right? So since the fish was collected largely around the main Hawaiian Islands, the specimens that I had, all the otoliths from the different size fish, I had small fish up to the biggest fish. I selected a series of small to big ones, and then I measured the radiocarbon content in those otoliths. And they were all from around the main Hawaiian Islands. So I stuck with just this one record from Kona as a reference for the ear bone. So what I do is I go in and I make a little template. Looking at the 2-year-old, I say, okay, where's the first year's worth of growth? Then I tap it out with a micromilling machine, which is a dentist drill bit controlled by a computer. It's like your worst nightmare. It's like the worst trip to the dentist ever. It's like 20 minutes of so it takes quite a while to extract each one, but I end up with like a couple of milligrams worth of material. Here's what it looks like. So see the little pile of powder right there. Okay, so a little pile of powder that's come off. And usually what I have to do is I have to kind of grind off the top of the otolith, and I look at the increment structure, look at the concentric, and I say, okay, there's the center. I want birthday stuff. Because what's in the middle is as old as the fish. This isn't like a normal bone where like our bones are turned over. No matter how old you are, your bones are probably 10 or 15 years old. These are conserved structures. They stay with the fish. So the stuff in the middle is as old as the fish. So that's what we're after. So try and sample within the first year, and then you typically get 1 to 3 milligrams, single sample, graphitization, ionization at the target, and basically it's all run at our friends at Woods Hole Oceanographic Institute at their NOSAMS facility, which is an AMS facility. So this is accelerator mass spectrometry that's actually used to come up with these numbers. They used to have to rely on the actual radioactive decay of carbon. Radiocarbon, and that was really tricky because it's a low energy, the margin of error was really high. So instead, now we're counting atoms. So back to the curve. Here's the fish data that I got. So each one of those squares is an extraction from a fish. Now the squares don't belong there. That's just the end point. That's when the fish died. So I know that the time span between that level and the core, which is birth year material, should line up with the curve. Now, if I add the counts, so what I'm doing is I'm taking the estimates from otolith counts and I count back in time. So like with this fish right here, I counted back in time to its birth year from the growth zone counts, and look at that, it lines up. So that means it's accurate, right? And pretty good too. And you notice from these teenagers here up through the 20s and 30s, it's pretty well lined up. Maybe a little bit of undercounting on this one. And this is actually a replicate sample. So core and then like a second core on that fish. Okay. And then the same here. So maybe a little bit of undercounting there, but largely we're on track. So the growth increments that we're counting in the sections are valid. And that way we can go in and we can do a whole bunch of fish and then describe the valid growth. Okay. So the take home message is that this species lives to be more than 50 years old, which is a little bit of a surprise. Did that surprise you? That Onaga lived to be 50 years old? Yeah, most people think, I'm a teenager, 20-something maybe. But yeah, a lot of these fish, and they get big quick. So they'll get big quick, but then they stick around a lot longer. Kind of like us, we get to a certain size, right? And then you die depending on how badly you treated yourself. Okay, so here's the Blue Marlin. So we're going to come back to this. This is kind of a figure that I made. So it's got the core is right here. So that's the stuff that I was extracting with the micromill. So I want the early stuff here. It grows up in this direction, sort of a horseshoe. So there's the ventral side and the dorsal side. This is called the ventral arm. And so in the sample that I'll show you, the very first one I did, it was actually from this earliest growth, which is the first year of growth. So this is again, it's a 1,245 pound fish, 3.7 meters. And co-author Jeff Sampaga, he is otolith remover extraordinaire. I mean, these fish have 3 sets of otoliths. They have 3 pairs. So they have the sagitta. These are the big ones, 8.1 milligrams. Then there's the lapillus and the asteriscus. They're even smaller. He found all 6 otoliths for this in a 60-pound head. So that's him extracting. Bob just found this. So Bob Humphreys is my former boss with Life History Program. He found this picture from 2009 when Jeff was extracting. And so you can kind of see that, you know, it's a little bit macabre here, but there's the eye, and he's getting into the brain cavity and finding these little teeny tiny things. And you can imagine if you just kind of mess something up, the otoliths are just going to disappear into the mess, right? So he got all 6. So this would not have happened. This validation of this particular fish would not have happened if Jeff hadn't found those otoliths. So with Onaga, we know where they are at a certain depth and around the islands, and we know that they're not going way out away from the islands. So we know their distribution, so we know the reference that we should use, right? So with the Onaga, I use the main Hawaiian Islands reference curve for the otoliths that were collected in the main Hawaiian Islands. But with Blue Marlin, where are they? Where did they start life, right? Where were they for the whole time that they were alive? Well, it turns out we know quite a bit about them, and that they are the most tropical of the marlin species. So they really like warm water, and I'll show you a little more information about that. But check this out. All of those places that are located here, so Curie, Hawaii, Palmyra, Rarotonga, Nauru, Palau, Guam, they all kind of cover the broad area of where marlin move around. So I thought, well, let's look at the radiocarbon signal across the board. Are there constraints? There's probably going to be some. It's not going to be as clean as with the Onaga, but maybe there's some constraints and we can actually age this really big fish. So here's what some of the records look like. So notice there's differences. So here's the pre-bomb, okay, 1950. The rise in radiocarbon is really steep for Curie Atoll, but then when you come all the way down to Nauru, there's some upwelling effects in this area. And so look at this. See how it comes up much more slowly and later. So, oops, and I accidentally went to the next one. So there's going to be a range of values depending on what I measure in the core of the otolith. So I just kind of separated these out so you could see the differences across the locations. Now, if we join them together, this is what they look like. So it's kind of a crazy, chaotic mess, but there are constraints to it. Okay, so it depends on how well the radiocarbon measurement from the core. If it's up there in the peak area, well, you know, I'm probably not going to be able to do very good with it. Maybe it's in one of the more diagnostic areas like this area here. Make this thing show. So this is narrower, right? So if I get a value in this area, then I'm going to know it's aged pretty well. So then, so I went in and gave it a try. So here's some other convincing information. So my friend Aaron Carlisle and others, he's at Stanford University in Monterey at the Hopkins Marine Station. He's been doing some tagging, and they found that that these fish really do stay in the warm water. I mean, look at that. They just avoid it like the plague. Look at here's a La Nina, and it's like they came up against a wall, right? They didn't even go. But then when it's warmer, they go to the South Pacific. So this is from Hawaii all the way down towards Fiji, I think, is somewhere down here. Yeah, some of them were released in Fiji. But during this La Nina year, they didn't go anywhere near that cold water. So that's a good sign. So that means that the otolith is uptaking radiocarbon to form the otolith. It's taking up that signal in that mixed layer. So I know it's going to be like the coral records. It's not going to be a deep signal because the deep signal can be depleted. They avoid low oxygen waters. So this is another paper that was recently done, Eric Prince and others. If they get over into these areas where deeper water is coming up, the low oxygen levels keeps them out. Not only temperature, but anything below, I think it's 3.5 or 3.7 milliliters per liter of oxygen, they don't want to be in it. They may go into it for a short foray, but they got to come back out because their metabolism is so high. I should say metabolic rate. So knowing that, what did we get from the carbonate? So the challenge is with this otolith, so I had to extract the core here and see where does the radiocarbon fall out. And I'll get to the other thing that's next to it. So here's the curves. Okay, so Curie is kind of the maximum that we could get for the range of blue marlin. It's kind of towards the upper extreme. They're not very common out there, at least it doesn't seem they are. Nauru is like the low one right in the middle. So here's the kind of the constraints that we can consider for the measured values. Guam is kind of right in the middle, and that exemplifies sort of the hot spots where marlin are found, which are North Central Pacific. And then as they come together, kind of put them together into a low S decline, So as they dovetail together, this can be kind of a monotonic decline that we can use to come up with some kind of age for a measured value. So here's what I got. So that is the otolith core on the collection date. So remember, like with the Onaga, I plotted it on the collection date so that as I project it back, I get the potential birth years. So check this out. There's the band of ages that are possible for this fish. So it's either on the order of 20-something, teenager to 20-something, or around 40 years old, one or the other. Now with just that one measurement, I don't know which one it is. I have no idea. Maybe the 20-something, because, you know, if you make conjecture about the increments that people were counting, oh, they're right and whatever, but we don't know with just that one measurement. So this other otolith that is right here, this is called the Lepillus, and thank God Jeff pulled this. What I did was I ran that one whole. So now, if it's the 40-year scenario, it's going to integrate the uptake of radiocarbon from this area all the way up through here, through this higher values, then down to the lower values when it was caught. So it was caught in 2009, so death date is right around here. It would have integrated the radiocarbon up all the way up through here. If it was just the young fish scenario, then it would only integrate the radiocarbon uptake from there, right? So it's an average value. The measured value I get from this whole Lepillus is the average uptake through the whole life of the fish. Does that make sense to you? So this is sort of troubleshooting. We're sort of narrowing down the constraints by doing some deductions. So here's what I got for that. It was right about 50 to 60%. So a dilution of 50-50 only on the decline makes sense that this fish was only 20 years old. It could not have been 40-something, because this value would have been integrated, would have been much higher, would have been closer to the core value or even above if it spent time up in this area. So I know that this fish, just with this one value, I know that that fish was about 20 years old. Actually, I know it's between 12 and 21, so the band is still pretty wide. So let's narrow that down a little bit. Now, another thing I did was I took the other arm, I sawed off the tip, and I actually did this under a microscope. I took a scalpel and I actually sawed through it very, very slowly for like 15 minutes so that it wouldn't break it off. And I got this nice little bundle of crystals. And I ran that. And that would either be 30 or 10 or 20, 10 or 15 years worth of growth, or it would be just 6 years worth of growth. And it turns out it's about 6 or 7 years worth of growth. That's where it would fall out as an average of that uptake. So now we have two separate lines of evidence from the same otoliths that point to the idea that this fish was about 20 years old. So let's take this a step further. Juvenile sagittal otoliths. Bob Humphreys collected some fish in 1996, and he got a few small ones, a couple of large ones, pulled the otoliths, perfect. So 1996, known age carbonate. These are otoliths. The numbers I'm going to show you next are from two fish that were 60 pounds. So we know that they were about, within the first year of life. So if I burn the whole otolith, that should be a reference mark for juvenile material. And that's like the core that I did for the adult. So here's what I got. Look at that. They fall right out on the line and slightly inside. So this intermediate scenario is probably more likely. Now I also ran a male. The males are smaller than females. They get to be about 2.5 meters long, maximum size. And they don't live as long, apparently. And it turns out, here's the collection date, and then here's the average value of the sagitta and the Lepillus, and they fall right in the middle here. So this tells me again that it's more likely that the largest fish, the grander, the 1,245 pound fish, was closer to 20 years rather than that 12-year. In fact, it's unlikely that it was over here because this fish was collected there. So it has to be some age, right? So the whole series of deductions that are narrowing this down to about an age of 20 years. Okay, so the fish was about 20 years old. They grow like a rocket, which kind of makes sense because they're still bringing in granders and they've been fished for a long time, so they've got to be growing pretty fast. So this is support for the age estimation techniques I showed you that people have been estimating from anal fins, dorsal spine fins, and the otoliths. So it corroborates their findings. The maximum age may represent what's being brought into the fishery. Fish greater than 3.5 meters are actually very rare in the long line fishery, and even in the recreational fishery. So this may represent the actual maximum age, but there's still a few large fish out there. So this is an example of a really large marlin. It's about 4.5 meters long. Choi's monster was brought in here to Honolulu also, which is a little bit surprising, in 1970. And that fish maybe is a little older. Maybe it's 30 or something, but who really knows, right?
How much did it weigh?
This, oh, sorry, that Choi's monster was 1,805 pounds. Yeah.
Like a car. Like
yeah. Yeah, it's like landing a rhinoceros on monofilament. It's crazy. Now, there's an interesting story about this. I'll get a little bit anecdotal here because we have plenty of time. So if you look at that fish, look at how thick it is. You notice it looks more girthy, and people have commented on that for years. And so since I was doing research on the largest, oldest fish, I just dove into it, and I started talking to all the people that had been making observations over the years. And one of the guys that is very well informed is Julian Pepperell. He's written some popular books, popular articles, but has done a lot of research on it's like, what was the actual weight of that fish? Where was that fish caught? Are there 2,000 pounders out there? And there's that elusive number that people say, oh yeah, 2,000 exists, but there's no proof of it. This is actually the heaviest that's ever really been documented fully. It turns out he talked with some other people, and I think he actually interviewed this woman right here recently. Get this cursor to work. Well, the woman that's in the picture on the lower right. He knew where she was, this was Jim Rizzuto, who publishes the Kona Chronicles. He talked to her, and it turns out that there was a tuna in the throat of this fish, 100 to 200 pound fish, and that's a bone of contention amongst people that were there. No, it was 100. No, it was 200. So whatever, it was a big fish, and it kind of looks like there was a tuna in the throat of that fish. I mean, look how girthy that fish is. And so if you take 100 or 200 pounds off of 1,805 pounds, that puts it down into more of a range that makes a little more sense relative to length. So the weight to length relationship tightens up if you do that. So it's sort of an interesting anecdote. And by the way, that fish was landed in, I think, it was like 2.5 hours, which is just insane for something that big. It probably would have got away. What they think happened was it tried to eat the tuna, and it got locked in its throat, and it suffocated. Because there's stories further. This woman said that they just took the boat over to the marlin and gaffed it as it was floating in the surface of the water. Yeah, so it was incapacitated and may not have been landed if it didn't do that.
You said that this catch was called something like
Choi's monster. Yeah, if you send me an email, I can send you there's a couple of cool little articles that I found.
How do they digest such a big fish when they catch that tuna? Is it just some digestive juices, some acids, or does it? Yeah,
muscular.
Like a cow where it brings it back up and chews on it.
Yeah, no, they don't bring it back up and chew on it. It's hopefully it stays down there. And in this case, I think the fins got lodged in the gills and it couldn't go back out. But yeah, normal digestive processes. Yeah. So let's see. So I've got another. We can either talk about more blue marlin, or I can go into a little bit of stuff I've been doing with lasers and the otoliths.
About the marlin, like
yeah.
A quick question. I went sports fishing with my son. I thought I'd show him, what I used to do when I was young. Not catch these big fish by any means. But I did manage to. I didn't. The guy who set up the rods on my rod number so and so. I was a lucky guy who climbed this 165-pound marlin. And the whole time I was fighting with it, and my son had to help me. I'm getting old. I kept asking the crew if they could let the fish go. Like they show in all the movies they show on board. They got this video, and the trophy fishing, they let them go.
Yeah.
And I said, no, it's over 100 pounds or something. It belongs to the owner of the boat, and you don't have any choice. It could maybe some initiative be started to change that, and maybe they let these fish go, or.
Yeah.
So money doesn't matter, you think? I feel sorry for the fish. I really didn't want them to see them kill it with these big metal bats.
Right.
It was terrible to watch it.
Yeah, there's quite a few studies about how these marlin, whether they survive or not after being pulled on for that long, and they actually, a lot of them die. I can't remember the actual numbers, but there's been quite a bit of study on catch and release success with this species. Yeah. So do you guys want to hear about lasers and otoliths, or should I just kind of leave it open to questions?
What was that otolith thing? Is that part of the spine?
So otolith is the fish ear bone. Sorry, you came in a little while ago. So it's a specialized ear bone. Yeah. So, okay, I'm just going to go through it kind of quick. So I made a discovery in 2014. I went to a Radiocarbon conference, Radiocarbon in the Environment. It was in Dublin, Ireland. I'm sorry, Belfast, Ireland. It was great. And I met this guy right here. This is Lucas Vacher. He presented on this laser ablation technique where they're using a UV laser to ablate the CO2, make CO2 come off of the carbonate directly. So you saw earlier that I use a micromill to extract a few milligrams. That sample is turned to graphite, and then that graphite's run on the machine. Well, they've got a situation now where they're running gas straight into the AMS. So accelerated mass spectrometry with gas. So it's really the next step in increasing the precision. So he was talking about his technology, looking for applications, and I said, hey, we've got otoliths. We could maybe get the whole bomb carbon signal. So rather than having just a single sample and kind of wondering where it belongs in the curve area, maybe we can do the full assay. So in steps red snapper. This is the true red snapper. Lucianus campechanis from the Gulf of Mexico. So I'm working with folks at the Southeast Fisheries Science Center at the Panama City Laboratory. They have huge otoliths, up to 4 or 5 grams. I mean, they're like this big. So the marlin otolith, you probably can't even see. You know, if I was holding it up, you'd have a hard time seeing it. But these otoliths are huge. So there's a lot of carbonate. So when you want to do a proof of concept, you kind of want to go with the most material, right? So I said, well, can we get this whole bomb signal going from the core, this is birthier stuff, all the way out to the edge? Can we run the laser across that? So I went and visited in 2015. This is the MyCaddis, which is the mini radiocarbon dating system. Oops. So here's this is Carol over here. So this runs through this instrument through a series of electromagnets, and then it gets, this is annoying, and then it gets counted in the machine. So this is the machine that counts the carbon atoms, carbon 12, 13, and 14. So here's the laser. This is the UV laser. The sample is in this little carriage. The CO2 that's liberated by the laser is pushed into the machine with a helium carrier. There's all these optimization techniques. So I spent a whole week there kind of working with them and trying to understand it and helping them to improve the design. So that was actually really fun. And so we've been moving forward with this. And so here's the otolith. These are the little tracks. So this is the cell, and so here's a laser track right here. This is the section, and I think I've got, yeah. So this area right here is this area right here. So we ran lines going from the edge back out to the core and then back to the edge again to see if we could see the whole radiocarbon signal. And it turns out we did. So to back it up, I had a core sample that told me, okay, this fish is definitely pre-bomb, which means it's before 1958. Now, if you notice on the coral curves, they sort of hit a flat line before 1958. So any fish that has a pre-bomb value, I can't tell how old it is. I can only say it's at least this old, could be older. So in this case, the collection date of 2004, pre-bomb estimate was 50 to 55 years. So it's limited to 1958, but it should have been born in 1949 or 54. So so here's the earliest counts, which are pretty easy to do. There's about 10 growth zones there. And so if we try and calculate where the rise should be, it should be somewhere in the first 3 to 8 zones. So I ran a series of ultra micro samples. These are really, really small samples to corroborate the laser results, which will be on the other side. And so here's the 3 tracks that we did to see if we could get it. A couple of just real short survey scans. We were just trying to see if we could get the signal. And it turns out that the radiocarbon signal, the rise, began more around 10 or 11 years. So it actually bumped the age of this fish up to about 56, 57 years. So it's actually much older. So here's what we got. So here's the coral reference record that we compare it to. The pink dots, the circles, are the subsamples I did with the micromill, and the other high error bar data, that's from the laser. So it's still very coarse, but we're getting the radiocarbon signal. So it's a first step in being able to use laser ablation to age fish. So yeah, 10, 11 years, 56 years. So it's very effective. So now we developed it. So I don't know if you noticed, but we had a rectangle that we were doing a zigzag across, and that crosses a lot of growth zones. So I asked them, can we do a parallelogram? And it sure as heck is true. We can actually do that. Get a little parallelogram. This is some of the dimensions. So it's only about 7 millimeters by 2 millimeters wide. Apu-upu, so we're going to investigate some of the really old fish that I have that may be 50, 60, or even 80 years old. Similar design. So the idea is that what brought this on is maybe as the technique gets better, we can start to do something like this with really small samples like the Blue Marlin. So is it possible that we could take this zigzag pattern right here and do that on the whole otolith, so we can get a better idea of what the life history is of these particular fish. And let's see, oh yeah, that's right. I presented this in just 3 weeks ago at another radiocarbon meeting in Hungary. And thank you very much. That's it.
Any questions for Dr. Allen?
So if you project forward 5 years in your career, what do you foresee this laser ablation technology doing for you, assuming it's going on a track that will?
Yeah, it may be the more effective way to analyze the otolith. So instead of going in and trying to tap out a micromilled core, it may be just prep the sample and then run a line across it and then calibrate it relative to the coral curve. So the cost may be lower and more efficient, and it's going to be potentially more time specific.
The error bars were from what though?
So as the laser's going across, it's blowing off CO2 gas, and so each one of those increments, so they relate the location to the timing on the instrument. So you have to kind of do an averaging. So it's really messy. If you look at it, it just goes like this, and then there's the bomb peak, and then it's like this. It's just this crazy mess. But if you take an average of all those samples, yeah, you start to see, okay, the precision's actually better than it looks. Yeah.
Just a question.
Yeah.
Choi's monster and the Blue Marlin.
Yeah.
They're females?
Yeah, they're all females. Yeah, the males only get to be 2.5 meters at the most. So that 3.7, that all the females have a completely different growth trajectory. Yeah. And there's all kinds of tails that they may get to be 2,000 pounds. There's some old Japanese records, and there's a photo that Julian was telling me about. He says he saw it, and it just looked much bigger than Choi's monster, and they can't find the document. It's in some obscure thing that they can't locate. So 2,000 may be the upper limit, but like gotta forget the number. There's one that's in Baja. There's a big cast Blue Marlin, and it's supposedly one that was caught on a long line fishery off of Brazil, and that it was so big that they had to weigh it in 3 pieces. They had to cut it in 3 pieces. But somehow there's no photos of any of this, right? It's like, and this was in the 80s. It's like I'm pretty sure somebody would show up with a camera for something like that. So it seems to be a bit of a fable, but it's pretty interesting when you start diving into the stories of the biggest fish, especially the ones that got away. Yeah.
I mean, everybody's talked about the older moments. But is there anything else in a bony fish that can bring about an age guess?
Yeah. So with the Blue Marlin, the dorsal and the anal spine also have growth zone patterns. They're a little more tricky because they get pulpy in the middle as they get bigger. They remetabolize part of the spine. So you have to make some assumptions about growth and sort of interpolate, right? But the fin spines can do that. With other teleos fishes, there's a lot of different structures. Fin spines in general tend to be used quite often. The operculum, sometimes they'll have a band structure. Scales for really fast growing fish. With long-lived fish, scales get remetabolized, so they get to a certain size, and then they're not conserved with time, so you can really underestimate age with scales on some species. Vertebrae with sharks, so that's elasmobranchs, so they have little tiny sand grain type otoliths called statoconia. And the vertebrae seem to be the best thing to use there. So you section the vertebrae and you get this little butterfly shape. And so you've got the core going all the way out to the end. You can count the growth bands in that structure. And sometimes it's accurate, sometimes it grossly underestimates age because they stop growing at some point.
So sharks are cartilaginous. So
Yeah. So they're still there, but the vertebrae, since it's such an important structural component, they do get calcified to a large extent. So it's not just, some species are pretty gelatiny, like whale shark and the Greenland shark, but most, like white shark, sand tiger shark, sandbar shark, they have pretty well calcified vertebrae. And you can see the zones in it.
Has anybody besides me seen the shark that's here in Triangle Lagoon?
Is there one here right now?
Oh.
Oh, you mean the little white tip?
June 9th.
The little white tip, there's a couple of white tips that hang out.
White tip, the water was so murky, I kind of went by me like a train, just maybe
Yeah.
2 feet from me, and I remember I tried to reach for my camera, I didn't have it, I had to go back to shore, grab my camera from my beach bag, and catch up with the shark a second time. And then I finally took some pictures, but it looked like they didn't come out until I started using some filters and increasing the contrast.
Yeah.
Using a red filter, I got a silhouette. I have it with me that you can see the whole outline. I'll see if I can find it easily, and maybe you can tell me what your guess is. But
Yeah,
you
there's
can't
little
tell
visitors
exactly
everywhere.
from that picture.
In fact, all of you have probably been seen by a tiger shark, but not seen the tiger shark. They're out there. They're not interested in you typically. They want sea turtles.
Any other questions?
Any other questions? Yeah.
Yes, sir.
Are you surprised by the age of these snappers of smaller fish from your analysis?
The age of the snappers?
Yeah, getting 50-year-old fish.
Yeah, I mean, it didn't come as a big surprise to me because I worked in the Pacific Northwest before I came out here, and so I was dealing with rockfishes. And those deeper dwelling, colder water fishes, like the yellow eye rockfish, gets to be 120 years old. And we validated that with another technique called lead radium dating, which is a radiochronometer that can validate their age, and they get to be 100 years old. Orange roughy get to be well over 100 years old. So when they said 50 for snapper, I was like, yeah, makes sense. Yeah.
So if you did statistics on how old some of these fish are and how many of them, because they're so old, they keep reproducing. Does the age coincide with how many fish there are?
Like the productivity of each
Yeah.
individual. Yeah, so it's called fecundity is like the ability to put off offspring, right? The number of offspring. And in some cases, like with the rockfishes, like I was talking about, the older they get and the larger they get, the more eggs they produce or larvae they produce. So it's interesting. Some species don't seem to show any signs of senescence. It's like, oh, this one's kind of old and it's slowing down. That just kind of doesn't seem to happen with most fishes, which is interesting. Yeah.
See, then if we overfish a species like that, it'd take a while for them to come back.
Yeah. So like orange roughy, yeah, it's hindsight 2020. Malcolm Clark did an estimate on if we had just found that fishery, like the one on the Chatham Rise or in other areas, to fish it sustainably, we would only be able to take 1 to 2% of the original standing biomass every year. We did a little more than that.
It takes a little.
Boom and bust. Yeah. Yeah, and that's what you see now. So if you see an orange roughy fillet that's like this big, that's juvenile, hasn't even had a chance to reproduce. Shouldn't be sold.